Welcome to Future Talk. On today's program, we're going to talk about recent advances in the biological sciences and some of the ethical and legal questions raised by the new technology. My guest is Hank Greely, Director of the Center for Law and the Biosciences at Stanford University. He's also a professor at Stanford Law School and a professor of genetics by courtesy at Stanford Medical School. He is chair of the California Human Stem Cell Research Advisory Committee and holds leadership positions on other panels and commissions, too numerous to mention here. He specializes in the ethical, legal, and social issues arising from the new biomedical technology, especially genetics, neuroscience, and human stem cell research. Hank, great to have you on the program today.
Pleasure to be here. Thanks for asking me.
So let's talk about genetics. We now have the ability to edit the human genome. What are the possible benefits we can get from that, and what are the possible pitfalls as well?
You know, actually, we've been able to edit genomes for over 40 years, but to me, it kind of reminds me of cars. There have been cars around for a long time. The first cars were really expensive. They broke down all the time, and almost no one could actually afford to drive them. And then Henry Ford came through with the Model T, and suddenly everybody could drive a car, and pretty soon everybody did drive a car. California and the world changed completely. What's changed in gene editing is a technique called CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is why we all call it CRISPR, invented mainly, I think, at UC Berkeley by a Berkeley professor named Jennifer Doudna in 2012. It's the Model T of gene editing. It has made gene editing fast, accurate, cheap, and easy. High school students with a garage and $1,000 can start editing genes now.
Now, how do you edit a gene? Do you go into a specific cell, find a strand of DNA, find a gene you want to change, pluck it off there, and somehow put another gene in its place?
It's not quite that. It doesn't follow quite that pathway, but ultimately that's what happens. You use this mechanism, this CRISPR mechanism, which homes in on a specific sequence of DNA. So DNA is a long set of letters, and all the letters are A's, C's, G's, or T's. So you could tell CRISPR, find a place where it's A, 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 T, 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 G, G, G. CRISPR will find that. It will cut it out and replace it. It's very much like a word processor's cut and replace function. And so you can do it in one cell, you can do it in lots of cells, you can do it in a petri dish, you can do it in a living organism.
Do you have to puncture a cell to get it in there, or do you just throw a bunch of genetic material at a bunch of cells and they're somehow absorbed into the right place?
There are different ways to do it, but normally it will be absorbed into the cells. Part of it depends on whether you're trying to do it in a real person or whether you're trying to do it in a petri dish. Let's take something like, say, insulin-dependent diabetes. We've got 35 trillion human cells in us. Some of us have bigger cells than others, but we all have about 35 trillion human cells. Most of those don't make insulin. Only a small number of them make insulin. For that, you'd only want to change the cells that actually make insulin, the beta cells in the pancreas. And you have to do tricky things to try to change only those cells and not other cells. On the other hand, with, say, blood tumors or blood problems, you can take bone marrow out, put it in a petri dish, change it in the dish, and then put it back in a person.
So is this actually being done now on real people?
Yes, not yet clinically approved by FDA, but in clinical trials with CRISPR. This is a form of human gene therapy, of treating people by changing their genes, treating people who've got genes that don't work by fixing the genes. It was first tried in humans almost 40 years ago and didn't work. It was only in 2017 that the FDA first approved a human gene therapy using some of the older methods. Now FDA expects to approve 10 to 20 gene therapies a year for the next decade, and most of them, I think, are going to be using CRISPR.
Well, I understand that a Chinese researcher recently did a CRISPR experiment which changed the genomes of a very small child and aroused a lot of controversy. What was that about?
Right, so there are lots of different ways and times and places when you can change genomes. If we were to say, try to change the DNA in your blood cells. Let's say you have let's say you've got cancer, and we want to change your immune cells so that they attack your cancer. There are new therapies out there that are doing just that. Most of them aren't using CRISPR yet, but they probably will at some point. We could do that. That's what we call somatic cell gene therapy. It's changing only your somatic cells, which are the cells of your body. When it works and you live another 30 years, but when you die at the end of the 30 years, that DNA change dies with you. If, on the other hand, we changed you when you were a fertilized egg, a single-celled zygote, every cell in your subsequent body would have that change, including the sperm, or if you were a woman, the eggs that you made. That could be passed down from generation to generation. We call the eggs and sperm the germline, and so that's called human germline genome editing as opposed to human somatic cell or body genome editing. It's a lot more controversial because it can pass down from generation to generation. That is what, in November, Hu Zhongqi in China announced that he had done to the embryos that became 2 baby girls.
And well, first of all, did the therapy work? What was the purpose of changing the genome?
Well, this is one of the most reckless experiments I can recall. Almost no one in the world has approved this experiment. It's been condemned almost universally, in part because there was nothing wrong with these girls. Or let me rephrase it, there was nothing wrong with the embryos that subsequently became these 2 little girls. He took a normal gene called CCR5 and changed it so that it wouldn't work anymore. Now, his theory for that is HIV, the virus that causes AIDS, usually needs or often needs a working copy of CCR5 on white blood cells in order to infect them. So his thought was, if I can make their white blood cells not have CCR5, they'll be exempt, they'll be immune from HIV infection for their lifetimes.
And what would be the problem with that if it were successful?
Well, the problem with that is, first, how likely are 2 baby girls in China ever to get AIDS, ever to be exposed to HIV? If they are, and it's 20 years from now when they become sexually active, how good will our preventions and cures be at that point? The benefits there look relatively low. But on the other hand, the risks of the first time you do anything in humans, and this was the first time anybody had ever made babies from gene-edited human embryos. Anybody who does something for the first time in humans, the first in human use, you need to throw salt over your shoulder, cross your fingers, find 4-leaf clovers, do all the Lucky Charms you can, because no one can really know whether or not it's going to be safe.
But it's going to be done eventually anyway, isn't it? Most likely, and somebody will have to be the first.
So it may be eventually done. It probably will eventually done, but the first time you do it, it's not going to be to give somebody some protection from HIV. It's going to be to change an embryo that would otherwise have Tay-Sachs disease, a deadly early onset genetic disease, or cystic fibrosis or sickle cell anemia. It's going to be take somebody who's got disease causing genes and change those genes to make them healthy. What Dr. Hu did was take normal healthy genes and change them in order to provide a protection. And what makes this worst is he didn't even successfully change them the way he expected. And even if he had, it would not have been complete protection. So this was an awful experiment for all sorts of reasons, most of which actually didn't really have to do with the fact that it was a germline modification. Most of it was just bad experimental ethics. But he became, he wanted to become famous in the worst way, and it is in the worst way that he became famous.
What about changing the germline in order to supposedly improve people, make them bigger, stronger, smarter, faster? What would be the problem with that if everyone's saying, well, I want genetic modifications on my baby, so my baby is better than any other baby?
Right. So we already know that grandparents think that their grandbabies are better than anybody's grandbaby. Parents have a way, and grandparents have a way of believing that, whether it's true or not. People are very concerned about these issues of enhancement. The interesting aspect of this with respect to genetics is we know almost nothing about genetic enhancements. we know literally hundreds of genes that strongly affect intelligence. When they are broken, you have extremely low intelligence. We know basically nothing about genes that increase your intelligence.
So just messing around in the genome is just as likely to cause catastrophic effects as the good ones you're seeking?
Catastrophic effects, or maybe slightly bad effects, or maybe significant bad effects with good effects that don't come through. Let's go back to the HIV example. The gene that he changed is found in this mutated form or an amutated form in maybe 1 person in 1,000 around the world, 1% of people in Northern Europe. It does seem to provide them with some but not complete protection from HIV, but there's evidence that it increases the severity of influenza or West Nile virus with them. So their genes do lots of different things. They're going to be pluses and minuses. The fundamental problem with enhancement at this point is we just don't know how to do it genetically. We know lots of ways to enhance people non-genetically. Feed somebody a good diet with a lot of protein as a child, and they'll be taller.
So there's some kind of regulatory agencies that says you're not allowed to do these experiments, but you can do these experiments. So is somebody in charge of this to make sure? And how do they have an abundance of caution? Are they more likely to err on the side of caution?
So one of the problems is all this is governed by national governments. There are 200 countries in the world with 200 different regulatory regimes. Now, most countries are not going to be able to do cutting-edge genetics, but quite a few can. Different countries have different sets of rules. In the United States, you could not do what was done in China unless the FDA allowed you to. The FDA will not allow you to unless you've shown the FDA good reason to think that this is both safe and likely to be effective, which no one will be able to do yet, anyway.
So how do we weigh the trade-off between safety versus possible benefits? How much risk are we willing to take in order to achieve how much benefit?
So I teach law students, and I tell law students that the first 2 words to any answer to a legal question should always be, it depends. Because it always depends. So for example, if you have a cancer and you're a competent adult and you say, this is a really risky treatment, but there's some reason to think it will work, FDA is more likely to let you choose that because you're a competent adult, your options are limited. This is a not crazy risk. If you're an embryo, you've never given consent to anything. You can't give consent to anything. Depending on how risky it is, maybe we take your parents' consent seriously. If the parents just want a child who's taller or a child who's got brown eyes instead of blue eyes, the FDA is not going to put up with a lot of risk for something with very little benefit. So in the US, we've got the FDA making these discretionary judgments, except a few years ago, Congress decided it wouldn't even let the FDA make those judgments with respect to germline, generation passing down genetic changes. The appropriations bill governing FDA says FDA is not allowed to even think about approving any of this. Now, appropriations bills only last for a year, so it'd have to be re-upped every year. So far, it has been for 4 years. In most of Europe, this is just flat illegal. China was unclear, and I think that's part of why it was first done in China. But as soon as it happened in China, Beijing decided, no, this violates our regulations. We have a regulation from 2002 that says it's illegal to do unethical and immoral things with human genetics. Now, in the US, would we necessarily think that meant germline modification? Probably not. But in China, when Beijing decides something is illegal, it's probably illegal.
So do you think that there might be a fundamental reason to say that we'll never want to modify the germline, or maybe when our understanding of the genome gets a little better and we think it's a little safer, then we'll feel a little more free to modify the germline?
So this is a hot debate. There are people who take this needs to be a line in the sand that we shall not cross position. And there are people who say, well, once it becomes safe and well known, maybe we should think about it. One of the things I think is interesting is, in fact, almost everything you would want to do through editing embryos' genes or fertilized eggs genes, you can do by a different method that's been around for at this point 29 years, called pre-implantation genetic diagnosis, where you make several embryos from a couple, you do genetic testing on each of those embryos, and you pick the ones that don't have the disease. You don't change the ones that have the disease so that they won't. You choose the ones that won't have the disease. That will take care of almost all the genetic diseases out there, but there are a few exceptions. So let's say you've got a couple where both the husband and wife have cystic fibrosis, and they're healthy enough and they've lived long enough, which today is possible. The life expectancy in the US for CF is about 40. 20 years ago, it was about 15. They want to have kids. Any child the two of them had from their own genes would have cystic fibrosis. They might be a good case for doing this germline modification so that they could have a healthy child. But there aren't going to be very many people in that situation.
Now, another area that you're very involved in is neuroscience, studying the brain, the nervous system. What are some of the ethical or legal issues involved in neuroscience research? What are some of the top controversies there?
Well, sure. I think the first thing to remember with respect to neuroscience research, people aren't paying billions of dollars from government agencies or pharmaceutical or biotech companies just so my friends who do neuroscience can learn cool things. We're doing it in order to try to understand better, prevent, treat, even cure mental illness and neurological disease. About 1 person in 7 on this planet, so over 1 billion people, have a mental illness or neurological disease. It's a huge source of human suffering, and we have not been very successful in the last few years in dealing with it. So there's a strong moral drive to do something. But the problem is, if you learn enough about how brains work to try to fix them, you also learn enough in order to do other things with them. Maybe control them, maybe read them, maybe modify them in ways that people don't like. If we could learn, say, to keep someone from being schizophrenic, from having false delusional thoughts, well, maybe we would learn how to plant delusional thoughts in them. Maybe, without using torture and old-fashioned mechanisms, we could truly use neuroscience to make someone love Big Brother.
Well, propaganda science is pretty advanced anyway. The art of trying to get people to believe something, that's been followed by many people for years. Does that take neuroscience to do that?
So I think that's a great question, because you point out that neuroscience isn't a completely unique and new way to do things. It's a way to do things presumably better than we've been able to do, but things we've been trying to do. Advertising, hype, it's what our society runs on. If neuroscience, though, allows us to do that more powerfully, more accurately, that could have concerns. So, for example, I'm trying to read your mind. We read minds all the time.
So it's a total blank, right?
No, I'm seeing things. You're wondering where I'm going with this, I can tell. We read people's minds because it's a survival skill. It's been a long time since we've had to worry more about lions and tigers and bears than whether the big guy coming at us is going to help us or going to hurt us. And we do it. We're just not very good at it. We can read minds, but not all that well. If we could read minds a lot better, there'd be some upsides. So if you can no longer speak for some reason, we might be able to figure out what you're thinking. There could be some downsides, too. If what you're thinking is, boy, I really don't like the government, that may not be something that you want the government necessarily hearing.
Now, in neuroscience, do we make a distinction between the hardware, which is the physical brain, and the software, which we might say is the mind?
So this is one of those deep questions that people go back and forth on. Most scientists, in fact, I think effectively all neuroscientists, think that the mind is something that is created by the brain. So think about a television set. Think about somebody watching this show on a television set. The television set is the hardware that's showing it. The electrons that are, the words we're saying, the meaning we have, that's the software. The brain is the machine that produces the mind. But if you change the brain, you change what it produces. So it used to be thought that there was a sort of dualist thought that our minds were somehow immaterial, separate from our body, not part of our brain exactly, but outside. Neuroscientists don't believe that anymore. They think the mind comes from the brain. The mind is what we care about more, but if you can change the mind by changing the brain, and as we learn more about changing the brain, which we're doing not really in order to read minds or to do brain control, we're doing it in order to cure Alzheimer's and in order to cure schizophrenia and depression. But as you learn more about the mind in some ways, you learn more about the mind in other ways, more about the connection between brain and mind. And so the possibilities for interfering, for reading people's minds, for making them do things, get stronger. We don't yet know how strong they get.
Has neuroscience come up with any solutions to any type of mental illness yet?
Not yet. We've come up with a
Got any good leads?
Deeper understanding. For example, we've learned more about addiction and some of the brain pathways and circuits that are involved in addiction. We haven't yet been able to put that knowledge into a great way of stopping it. We know what happens when people get Alzheimer's disease, what kinds of cells die where. We have some guesses about why they're dying, but we don't know enough yet to be able to stop it. But we are learning. The brain is amazingly complicated, but I don't think it's infinitely complicated. And as we learn more, we will ultimately be able to intervene in some useful ways. Unfortunately, in some respects, ways that we're learning right now aren't particularly medically useful. So there's an area in your brain back here that lights up, that gets more blood flow when you see a face, or when you see a full moon, because you perceive a face in the full moon, when you see something that your brain thinks of as a face. So 20 years ago,
lighting up means more activity, the circuits are more activated. Activity.
So it's actually sort of an interesting mechanism. Your brain is the energy hog of your body. Your brain is about 2% of your body weight, but it uses about 20% of your energy. And when an area of the brain has worked, shortly after it's worked, 2 to 4 seconds after it's worked, it gets flooded with fresh blood carrying fresh oxygen. And so we can use a tool called functional magnetic resonance imaging, which is your basic MRI machine, but instead of looking at structure, it's looking at blood flow, basically. And we can say, the fusiform face area in Marty just lit up. That means 2 to 4 seconds ago, he saw something that his brain thought was a face. That's pretty interesting. I can tell you, Marty, think about moving either your left hand or your right hand. Don't tell me which, but think about moving one but not the other. And by looking at this fMRI, I can decide which hand you plan to move, because there's an area called the secondary motor area that plans your movements. And one of them's on the left side. The one that controls your right hand is on the left side, and the one that controls your right hand is, your left hand is on the right side, because brains are confusing. But I can tell which hand you're planning to move. Now, I'd much rather be able to cure you of schizophrenia or depression or Alzheimer's disease. We're not there yet,
now,
but that's the direction we're moving.
Isn't there also research in your mind controlling physical objects, like a prosthetic hand, for example,
Yep.
using brain impulses to move the fingers on an artificial hand?
And that's a great example. People are doing these brain-computer interfaces in order to help people who are quadriplegic move and control things, and it works. There are people who are able to send emails by thinking about particular letters. Now, right now, they get about 8 letters per minute, which is slower than most of us can type, and it costs hundreds of thousands of dollars, and it doesn't last, they can't do it for very long. So it's a proof of principle. It's kind of, though, to go back to gene editing, we need the Model T. We need the thing that will take this from being really expensive and really tricky and really difficult to cheap and easy. And that's where neuroscience is kind of hung up right now. We're trying to make the move from expensive, difficult fMRI to something cheaper, easier, more accurate, faster.
The neuroscientists feel they're on the right track. If they just keep pursuing the direction they're going in, eventually they'll get some good results, or do they sometimes wonder if maybe they need a new direction?
So there are a lot of neuroscientists, and they don't all think the same way. But I do think neuroscientists in general would love to have better tools. The U.S. has a brain initiative. It's mainly from the NIH, but other government departments as well. And this brain initiative is actually spending several $100 million a year, mainly to try to make better tools to allow us to understand better and intervene better in people's brains in order to try to treat and prevent mental illness and neurological disease.
Now, some mental problems include things like excessive anger, which isn't necessarily classified as a mental illness, but certain emotions, they're good for survival under some circumstances, bad in others. Is neuroscience also tackling basic human behavior among people who are so-called normal?
Sure. We are trying to understand better how people react. One of the problems, though, is to understand a human brain, the best way to study it is a human brain inside a human body, but we humans are terrible lab animals. You know, we lie, we cheat, we don't follow orders, our generation times are too long, and we've got lawyers. I can't put you in a lab, try something, take your brain out, slice it up, look at it, and put it back. So we can't use people, we use mice, or sometimes we use monkeys. The problem is we know that we're not mice and we're not monkeys. There are things about humans that you really can't study in non-humans. And one of the really interesting things that's happening these days is scientists are trying to come up with models, with surrogates for human brains. So they're making mice or monkeys that have some human brain tissue in them. Or they're making little bits of artificial human brain, things about the size of a pea that have 2 to 6 million human neurons in them, and watching how they develop. Taking, say, cells from somebody with autism and seeing how those develop as neurons, or they're taking chunks of brain tissue from neurosurgery or from corpses and keeping those chunks, I want to put scare quotes around this, quote, alive, close quote, in order to study them.
I have a lot more questions I'd love to ask, but unfortunately we are out of time, so we're going to have to stop. I'd like to thank my guest, Hank Greely. Thank you for watching. Visit our website, www.futuretalk.net. For Future Talk, I'm Marty Wasserman, and we'll see you next time.